Good morning, everybody. Welcome back. So I've got a couple reminders, and then I'll go right into our program. So we're having breakfast. Help yourself. Remember that we have the door leading outside to our nice covered lake view. For today, we do have extension cords if you want to be charged. So they're in a pile over there. The thing about this old room is that it's not modern in the sense of having power outlets everywhere. They're kind of around the perimeter, but if you feel like you need something charged while we're here today, you might need to set it aside somewhere and either grab an extension cord or find so the extension cords are on that little cart, but the power outlets are kind of dispersed around the exterior. So just so you know what your options are, they're not really at the table, unfortunately. Travel questions. So you'll probably be getting updates today and so on. Like if anything changes for you and you need help, please see Mary Lynn on the back table. She's our travel guru. But from our perspective right now, everything's pretty set. So if you need to make changes, please see her. I think yesterday we had a really good overview of a lot of things. So we had some deep dives in All of Us and Ad Health, outlining some of the data and opportunities, and then quite a bit. So we had 14 presentations yesterday, so it's a whirlwind, but we had some real frontier research on applications of polygenic scores, further incorporation, family-based designs, and family-based proxy data in our family session, and several innovative takes on next steps with DNA methylation data to assess health and accelerated aging.
All right, welcome back, everyone. So we're going to continue talking about epigenetics and also get into some transcriptomics in this session. So this session is entitled Biological Aging and EWAS. And first, I'd like to introduce Dr. Eric Klopack from University of Southern California, who's going to talk about development of a novel transcriptomic measure of aging, transcriptomic mortality-risk age.
So much. Really happy to be here, really excited. We're talking today about a measure of aging based on transcriptomics or RNA data that we have been working on using the health retirement study data. So yeah, just starting off with a quick acknowledgement to funders, and mostly from NIA through various mechanisms. And a special thanks to Bharat Thayagarajan, Steve Cole, and Eileen Cremens, who have all been really helpful with this work. So generally speaking, there's this argument we call the geroscience hypothesis that aging is sort of a unified, coordinated, multi-system decline that happens across the life course. And so it happens at multiple biological levels as well as across multiple systems. And so a real big goal of the geroscience agenda has been developing biomarkers of this aging process that you're able to measure in people before they actually, get various morbidities or, die. And so trying to get measures of this process as it's happening has really been a big goal. And there's been some work on trying to develop transcriptomic or RNA-based measures of this aging process in the past. But that has mostly been developed using kind of small specialty samples. A lot of them used array data rather than full RNA-seq or RNA sequencing data. And they tended to use tissues other than blood. While at the same time, large population data sets like HRS, like Ad Health, like we heard yesterday, as well as like MIDAS, the Nicola, the Northern Ireland aging study, are collecting large sets of RNA-seq data, so this high throughput next-gen RNA sequencing data. And so we have this now enormous opportunity to ask, population-based questions, try to understand how population-based aging is working across the life course using cohorts that have different age ranges. But first, we really need a reliable measure of accelerated transcriptomic aging that works in blood, in diverse populations, using this RNA-seq data. So we already have a couple of other types of aging measures. We talked a lot about the epigenetic clocks, and we're going to talk more about those. So why might we be interested in RNA or transcriptomic data? So here's this kind of a cartoon example of how we kind of think that RNA might be involved in the sort of process by which social and environmental factors get under the skin to affect health and aging processes. And so social environmental factors have various signals that lead to different transcriptional factors deciding whether DNA is expressed or not. And so one of those would be DNA methylation, which the clocks are based on. RNA then, this messenger RNA is kind of the next step in between the DNA being transcribed into RNA and actually being expressed as a protein. So it is a little bit more biologically proximate to the actual outcomes we may be interested in. It is closer to the actual proteins that are going into leading to physiological decline. And so it's a little bit more proximal to the outcomes we're interested in. And it also may just capture other parts of the aging process that the clocks just don't capture. And so it's a kind of another interesting omic that we don't really have a good an aging measure for yet. So we developed this sort of novel transcriptomic aging measure using the health and retirement study. So I hope you've already mentioned or talked a little bit about the health and retirement study already, but it's a nationally representative study of older Americans, so age 50 and older. And we're using the 2016 venous blood study, or VBS. This was collected on about 10,000 people. About 4,000 people had RNA-seq. Actually, they're RNA sequenced. And of those, a number of them were dropped for sort of technical reasons, and then a number of them didn't have mortality data. So our final sample here ends up being 3,595. These are all community-dwelling older adults in the U.S. And then of these, nearly 3,600 people, about 407 of them died. And then here's just a little bit of information about how this was collected. So we used packaging tubes. A phlebotomist collected the blood. We used the TopMed pipeline to process the data. And I used log2 adjusted counts per million for all of these analyses. So to actually train this measure, we used ElasticNet, which is, we heard a little bit about yesterday, but it's a machine learning approach that is able to do both feature selection, meaning it's able to trim this large number of genes that we've captured into a more manageable set. It also helps correct for overfitting within our training sample. So if we just included all of them, the data would be way overfit to the actual sample that built it and would fit really well in our sample, but not as well in other samples. This does shrinks betas down using cross-validation to try to actually create a set of variables that best predicts the outcome and is generally going to be replicable across data sets, hopefully. So our ultimate outcome here is 4-year survival using a Cox estimator. So this is a time to death measure. So this would be in 2020. The 400 or so people died by 2020. And so this is the amount of months in between their blood collection and their mortality event. So in HRS, across all of the RNA sequencing, we were able to get data on about 50,000 genes. And then we also included in this measure age and sex as kind of confounder variables that we want to kind of adjust out, but also might be causally important in our model. So we selected the penalties, basically how much the machine is actually going to penalize all of our betas using 5x cross-validation and a grid search method. These are the two we happen to pull out. And ultimately, this machine selected 39 genes in chronological age as being kind of the important features here that are going to be useful for predicting mortality risk. We then evaluated this in a testing set. So we split the HRS data basically in half randomly. And then we trained this in the training set. Now in a testing set that is naive to this process, we used the weights from the training set and estimated the TraMA score for the testing set. And so here's kind of a little cartoon example of what the actual math looks like. It's really easy. You multiply each gene or age by the beta and then sum them together. And then to make this on a very easy to kind of interpret metric, we then transform this to have the same mean and variance as chronological age in the testing set. So the metric here is in years of life. So the mean here is kind of I know the color's kind of skewed here. It's purple, I promise. The purple vertical line, around 70-ish, 72, I think, is the actual mean age in the population, and it's the mean of our TraMA measure. As you can see, we get a fairly normal, though a little bit skewed distribution. So how well did this machine actually do? How well did we do in actually predicting our outcome here? So in the testing data, TraMA was very strongly associated with mortality hazard. So Harrell's C index, which is very similar to area under the curve, if you're familiar with that metric, 0.5 would be random chance, 1 would be perfect prediction. We get a 0.8, which is kind of on the borderline between excellent and good. That is sort of at the cutoff for excellent. So we're doing a very good job of predicting mortality hazard. And then just to make kind of a visible something to look at example, here we have actual just observed mortality as a dichotomous measure. So people who died had a TraMA age 11 years older than people who did not in the sample. And that is significant using just a t-test. But this is not really adjusted for anything, so it's a little bit going to be biased by the fact that age is included in both of these. So, because a lot of us are social scientists here, we're also really interested in how are these social and demographic factors associated potentially with this biological or this aging measure. So here, this is controlling for age as well as technical covariates. So TraMA is associated with greater educational attainment. So for people, this may be hard to read, but for here on the audience, house left, stage right, so for people that have 11 or fewer years compared to people who have 16 or more, they had about 2 more years of TraMA age. And similarly, there's a little bit of a stepwise pattern. As you get more education, you tend to have a younger TraMA age. Women appear to be aging a little bit slower than men, so they have a TraMA age about 2.2 years younger than men on average. And then the racial differences were not as clear as they are often in the clocks. So the only significant result here is for the other race, not Hispanic group. So these are kind of associated in general with these social and demographic factors, but it's also important that this be associated with health outcomes if that's ultimately what we're interested in finding out about, how do these demographic factors get under the skin to affect health. So we find that this TraMA measure is pretty strongly associated with some of the key health and aging outcomes that we're all interested in. So here with multimorbidity. So for each about half year of TraMA age older you are than someone else, you have one more predicted multimorbidity. You're at greater risk of mortality. People who have an older TraMA age tend to have poorer cognitive functioning, and they tend to have more disability as measured by activities of daily living and instrumental activities of daily living. And then we've done some additional research here that I actually didn't have time to put in the slides, but we have a little bit of mediation analysis that suggests that this measure does also mediate the association between these social variables, so between education and sex and these health outcomes. So to some extent, it looks like this is potentially a mechanism by which these social inequalities are becoming embedded and becoming health inequalities. So some limitations that this measure has at this point, this is all older adults, so they're all 50 plus. So this may not translate very easily into younger populations. It may not translate as cleanly into non-US populations since it was trained and then tested in all US cohorts. Also, RNA here was only collected at one time point. So ideally, we would have longitudinal measures of RNA and we could look at how things are changing over time. Kind of in a similar way that the pace of aging measure works, or the methylation pace of aging works. But we only have RNA at one time point. We do have longitudinal information on mortality, but not on RNA. So the future steps here, we got some really good replication within HRS, but the ultimate goal here is to have replication in other large cohorts. So we've been working with Nicola, the Northern Irish study, as well as some other studies to try to get replication with both mortality and then potentially in other age groups to see, does this actually work in other age groups? And I've got some very, very preliminary results. Again, I didn't put in here because they're very, very preliminary, but using gene expression omnibus data, people who died in hospital from some of these gene expression omnibus studies tended to have older TraMA age as well. So that's all I have. Thank you very much.
Eric, finally, we have Lauren Opsanick. Am I saying that right? Opsanick.
Close.
Who is a PhD student in epidemiology at the University of Michigan.
Good morning, everyone. Thanks so much for having me here. I'm really excited to share some of the work that I've been doing for my dissertation. So I'm going to talk a little bit about today an epigenome-wide association study of long-term psychosocial stress in older adults. So oftentimes when we discuss stress from a research perspective, there are several different types of stress that we can be referring to. So it could be physiological, biological, or psychosocial. And this study is going to focus specifically on psychosocial stress. So by definition, psychosocial stress is the consequence of an imbalance between adverse experiences that we might meet in our everyday lives and then our ability to cope with them. And so as you can see from this diagram, psychosocial stress can be triggered throughout the life course by a multitude of stressors, which include adverse life events like job loss, discrimination, health issues, or financial uncertainty, as well as work-related or community stressors. And as many of you know, and we've discussed a lot yesterday, high levels of stress are associated strongly with adverse health behaviors, which include smoking, increased alcohol consumption, and decreased levels of physical activity. Additionally, stress is linked to the development of certain chronic diseases, which include diabetes, hypertension, and heart disease, among many others. So in terms of the epigenetic impact of stress, there's been some previous research that has found associations between psychosocial stress and very specific candidate genes that they were examining. However, I've found that there's very few, if any, epigenome-wide association studies that have examined the relationship between psychosocial stress and the entire genome. So I know we covered this a lot yesterday, so I'm not going to go too much into this. But just for some quick background, epigenomics is a field of study that focuses on changes in the DNA that does not involve alterations to the underlying gene sequence, and instead marks are added or removed to regulate gene expression and change how proteins function. So we talked about how epigenetic modifications can be reversible, and they're also responsive to certain environmental stimuli, which do include alcohol consumption and smoking, which will be important for my some of my analyses later. And so DNA methylation, which is occurring at the CPG sites in the genome, is going to be the easiest measure of epigenetic modifications, and that's what I'll be using in this study. So there's two research questions that I'm really hoping to answer in this study. The first is I want to understand, after we adjust for all of the potential confounders in this model, is long-term psychosocial stress going to be significantly associated with DNA methylation in older adults? And then second, if we do observe an association between stress and DNA methylation, do certain health behaviors actually mediate this relationship? So as I mentioned previously, it's been well established that stress is associated with adverse behaviors. And then a lot of those same behaviors often impact DNA methylation. So it's really plausible that any of the associations we do observe between stress and DNA methylation are going to be mediated through certain health behavior pathways. So for this study, I used data from the Health and Retirement Study, or HRS, which we've now covered extensively, so I won't go into too much detail. But I wanted to mention that in 2006, HRS added a psychosocial and lifestyle questionnaire, which is a self-reported psychosocial survey that was administered to a random 50% of the total HRS sample at each biennial interview. And this survey was really interesting, and it was probably one of the most extensive surveys that I found examining stress. So it captured a lot of information regarding participants' well-being, their lifestyle, social relationships, and a lot about their mental health. So on the right, you can see a timeline of when all of the measurements were collected for this study. So my main exposure of interest was psychosocial stress, and it was examined in either 2010 or 12, depending on which year the participants were administered their psychosocial questionnaire. And then in 2016, DNA methylation from whole blood samples was collected using the EPIC chip. And for this study, we quantified DNA methylation using beta values, which approximates the proportion of methylation, And so after quality control, we were left with approximately 790,000 CPG sites in a sample of about 4,000 individuals. And then once we excluded all individuals who were missing any psychosocial data from not having their battery administered or any relevant covariants, we had about 2,700 participants who were included in our primary analysis. So I am not a social scientist, so I wanted to kind of follow the literature on what others have done with psychosocial stress to use as my kind of primary measure of stress in the sample. So consistent with previous research that studied psychosocial stress in both the health and retirement study, as well as other cohort studies, I examined 6 domains of stress, and those included acute life events, financial stress, neighborhood stress, relationship stress, which looked at specifically marital stress, stress with your children and other family and friends, and then lifetime discrimination and childhood adversity. So as you can see from this list, individual stress domains included 1 or more stress measures. And we did this so that we could get a really broad understanding of stress because it comes from so many different facets. And so we first standardized each stress domain by transforming them into z-scores to allow for comparisons across domains. And then because we were interested in cumulative stress, again, following the literature, we created a summary stress score using the 6 individual stress domains by summing the standardized scores for all of the domains and then re-standardizing those scores. So this cumulative stress score represents the total stress exposure across all of the 6 domains. So here you can see we have a table with our sample characteristics. I won't spend too long on this, but just wanted to highlight some things. So the mean age of our sample was approximately 70. We did have some variability in terms of race and education, with the majority of our sample having a high school degree as their highest level of educational attainment. And you can see on the bottom left, we had a very, very large distribution in household, total household wealth for the participants. And then in terms of health behaviors, it was a relatively healthy sample. So about 10% were current smokers at the time of their interview. 40% were moderate to heavy drinkers based on the NIH alcohol consumption guidelines. And then over half of the participants were considered active, meaning they engaged in moderate to vigorous exercise at least once per week. So in terms of methods, we first conducted an epigenome-wide association study using multivariate linear regression models to estimate the effect of DNA methylation at each CPG site. And we adjusted for age, sex, education, top 10 PCs of genetic ancestry, white blood cell proportions using a Hausman method, and then also technical covariates. So we considered this our primary model, which you can see up there, we labeled as model 1. And then we adjusted the p-values for multiple testing using FDR correction with an FDRQ of 0.1 as our significance threshold. And we then investigated the attenuation of effects for the significant CPG sites identified in our primary analysis by further adjusting for additional lifestyle factors like marital status, employment, having children, and then total wealth, which we refer to as model 2, as well as health behaviors, including smoking, alcohol use, physical activity, BMI, which is model 3. So then next we wanted to tease out that relationship that certain health behaviors may have had on the association between stress and DNA methylation. Therefore, we conducted a formal mediation analysis for all CPG sites that were significantly identified in our EWAS using the Carlson-Holm-Breen or the KHB method. And so one major advantage of using this KHB mediation method is that it allows us to examine multiple mediators simultaneously, which is going to be very helpful for us because we know a lot of these health behaviors can be correlated. So now getting into some of our results. So on the left up there, you can see is a Manhattan plot from our primary results. So the red horizontal line that you see represents our FDRQ of 0.1, which was our significance threshold in this model. So you can see that there are 9 CPG sites that have been annotated across the genome that are significant according to this threshold. And then on the right, we have our QQ plot looking at the observed versus expected p-values in our EWAS. So towards the top, you can see some deviation at the tail, which is indicative of p-values that are more significant than expected under our null hypothesis. So here's a list of the 9 CPG sites that were FDR significant, along with their summary statistics, as well as their functional annotation. So I know this is kind of a lot in the table, but based on the direction of effects, 8 of the 9 CPGs were actually less methylated with higher levels of cumulative stress. And then following the columns in models 2 and 3, once we adjust for additional lifestyle factors and health behaviors, there's really not too large of a change in the beta value, and the p-values are still showing a high degree of statistical significance. All of them, I believe, are less than 0 or 1 to the negative 4th. So then that leads into our mediation results. So that we found that after adjusting for all of our socio-demographic characteristics, including those lifestyle characteristics like marital status, having children, and wealth, stress explained between 0.5 and 5% of variability in the 9 identified CPG sites. And then stress explained between 0.3 and 3% of the total variability after further adjusting for health behaviors. And then when we assessed the mediation of the health behaviors simultaneously, we found that smoking, moderate alcohol use, and BMI partially mediated the relationship between psychosocial stress and DNA methylation. So in this table we have, you could see which CPG sites were significantly mediated and by what health behaviors, as well as the percent mediated. So we found that physical activity did not mediate significantly the relationship between stress and any of the 9 CPG sites. And my guess is probably due to the high correlation between other behaviors, particularly BMI. So it's clear based on these results that health behaviors are really only partially mediating the relationship between stress and DNA methylation. So what we found is that many of these CPG sites have been previously identified in other EWAS, and I have on this table up here. So our most significant CPG site was found on the RRM2 gene on chromosome 2, actually in the promoter region. And this is an important gene for DNA synthesis and DNA repair. So in large-scale EWAS, which I found really interesting, this gene has actually already been previously associated with schizophrenia and PTSD, which are 2 obviously psychosocial disorders that are related to our exposure of interest. And then several other CPG sites were found to be previously associated with things like inflammation, type 2 diabetes, cognition, and various types of cancer, among other things. And this is kind of what we were expecting to see. So in terms of significance, this is a novel EWAS that looked at psychosocial stress in a multi-ancestry population of nearly 3,000 older adults. So we identified 9 CPG sites associated with long-term psychosocial stress, some of which are partially mediated by health behaviors. So it's important to note that when these health behaviors are taken together, they really do explain a relatively low proportion of the relationship between stress and DNA methylation. So it is possible that stress impacts methylation through some independent pathways. And this is really important from a public health perspective, because when we oftentimes discuss stress, a lot of emphasis is put on improving our health behaviors, like increasing level of physical activity and improving diet in order to combat the effects of stress on negative health outcomes. And obviously that's extremely important, but this research shows that maybe there might be other pathways that stress operates through as well that do affect the epigenome. So this further supports the need for mental health resources, especially starting from a younger age, to manage the root cause of stress as opposed to kind of getting at stress after it's been already happened and manifesting. And then another thing to note is that if there is a link between psychosocial stress and DNA methylation, this relationship may be further intensified and further intensify disparities in certain racial and ethnic groups. So evidence has shown that racial and ethnic minorities have higher rates of psychosocial stress, and that's due to a variety of factors like discrimination and economic disadvantage, et cetera. And additionally, we found that minorities are often less likely to have the mental health support to help adequately manage their stress. So the impact of stress on the epigenome may actually be greater in certain racial or ethnic groups. And so this is something I'd be really interested in looking at as well. So in terms of next steps, since we do have some potential evidence that stress is linked to DNA methylation, I plan to evaluate whether DNA methylation then mediates the relationship between psychosocial stress and cardiometabolic factors like diabetes, hypertension, cholesterol. And this can really help to understand the mechanisms underlying the negative health outcomes, and particularly chronic diseases caused by long-term stress. So I'd like to acknowledge my advisor, Dr. Jennifer Smith, who's in the audience, and the rest of my colleagues at University of Michigan, and then Lauren at University of Wisconsin for all of their guidance on this project. Thank you.
Okay, welcome back. So next we are going to have an exciting session on gene-environment interaction. Our first speaker is going to be Dr. Evelina Akimova from University of Oxford, who's also joining Purdue shortly. And she's going to talk about gene-environment interaction analysis for chronotype and sleep duration traits among shift workers in the UK. Welcome.
Okay. Hey, everyone. And I want to use this opportunity first to say huge thanks for everyone who's been organizing this, actually. So thank you. And let's talk about sleep and a bit of jet lags as, asking for a friend. Let's start. So I was saying that I really liked the title of the session in terms of innovative ways. I don't have me. And I think one of the another branch of the work we've been doing as a team in Oxford, it's actually work on sleep, on chronotype, on sleep penalties, and on occupations and different types of occupations and contracts, because there, especially with COVID, as you all know, there have been lots and lots of changes. And so, as you know, apart from fertility and other things, we've been doing a bit of chronotype work. And today I want to talk about links between genetics of sleep and shift work, because it's, for me, another actually area where I think we as field can contribute to answer some very, very important sociological questions, because I'm also a social scientist by training, and I'm every time keen to learn what is useful to use genetic data. So as you might know, generally shift work is on one hand is actually growing in the UK and in the US. I think it's even up to 25% of people involved in shift work, and obviously it depends on the time you measure it in terms of birth cohorts, etc. But it also perceived to be as one of the risk factor for your health. And there are multiple ways why. I was surprised to learn that WHO actually considers shift work as one of these risk factor for cancer. So even for the questioners, you know, like if you shift worker, you go to different basically basket as you might be more likely to get cancer and other health problems. One of the mechanisms, though, that researchers have been linking why shift work has such adverse health outcomes is through sleep, basically through sleep disruptions, and specifically what's dangerous, it is prolonged sleep disruptions. So that was basically one of the motivation, kind of substantial motivation of our research questions. And on the other hand, I think, you know, we just live in the time we live, like 21st century, we're used to this from 9 to 5 schedules. You know, it wasn't like this all the time. It's just industrialization and 20th century when we as society shifted to different time schedules, different even school timings. So basically, that also, and another thing that inspired me to kind of why it would be interesting to look at these things as sociologists. Here, I just wanted to basically to provide you a bit of trends that those who involved in different types of shift work on average, they do sleep a bit less. This is our own predictions from UK Biobank, especially those who always work shift works. So, and specifically night shift works. Here you see, so generally there are sex differences, especially you might be aware that on average, I think men do sleep a bit less than women, and sometimes there might be some biological hormonal differences. But even for night shift work, we also observe this trend. So basically in terms of sex-specific differences, there are, and they're also important to consider. Another type of another factor actually that is important for this type of phenomena is length of working week. So those who tend to work long hours, even when they're always in night shift works, as you see, they do sleep even less. And while I was a bit torturing you with different types of visualizations, I wanted to note, as you see, there is sometimes, usually, always, these different types of groups, basically different types of night shifts. And I wanted to stress that it's not linear, right? As you can see, it's not necessarily just line, because if you look at the underlying occupations of these different types of shift works, they are actually very different jobs. And there are also different jobs among women and men. So that's why we treat them all categorically, no linearity assumed. And here I want to talk about kind of, because when you're interested whether there is heterogeneity of this like sleep penalties among shift workers, it kind of, within our field, it's kind of gene-environment interaction conceptual framework. But I want to stress that actually it's not what we preliminary, kind of theoretically, how we frame the paper, because we are interested in interactions, but for example, I'm not interested in whether there is some causal story, whether shift workers really suppresses some genes or not. I'm interested in very general, whether there are heterogeneities in something that I'm, as a social scientist, it's very hard to measure different types of chronotypes, etc. So basically here, I'm more proxying something that I usually don't have in the data, such as whether you're more evening or morning person through polygenic scores. So, and that's, and I'm happy to answer the questions, but that's why modeling-wise, it is gene-environment interactions, but what I learned from it, or what we learned from it, it's a bit different. And hopefully I will maybe convince you at the end of the talks that this is also type of things we can think and push a bit more. So we have 3 research questions, whether basically higher genetic score of being more evening person, whether it promotes greater night shift work tolerance. We also interested whether these moderating patterns dependent on the length of working week. And also what is exciting and challenging, we wanted to look at different sources of sleep data, because obviously when you report your own sleep data, you might be a bit like it's not the most reliable one, as you can imagine. So we used both wristband data from UKB and self-reported one. What we did, we looked at UK Biobank. So there are lots of pros and cons, obviously, in terms of generalizability, etc., and everything. That's why I'm not going to convince you that there is anything causal happening here. But what I want to stress that actually UK Biobank is a great resource to study night shifts and different types of basically shifts, because I don't know whether you worked with this type of job arrangements. Usually sample sizes are very, very small. Like at least in another data, like British household panel data, if you heard of it, I think there might be up to 500 people in each wave, so there is like, and especially they're not genotyped and they don't have sleep information. So UK Biobank is actually quite good and rich source to use. to at least look at shift workers. Basically, as I mentioned, we looked at different types, like information of sleep, and we did lots of exclusions. We were interested in those who are working, and obviously age brackets were also selection criteria. And here, just a bit of descriptives in terms of what I wanted first to show, that it does, UKB has the same trend as it was found in other data sets, that it's usually a bit more man involved in shift works than female. But overall, it's actually quite good sample size, and even like quite good number of those who involved in different type of shift works. So that's good to think about UK Biobank. And here, this is the comparison basically of wristband sleep duration information and self-reported duration of sleep. You see there is not that much differences, and the error bars for accelerometer data are kilometers, because it's much smaller sample size. Of those of you who work with UK Biobank, you know that there are only portion of people who were asked to wear wristbands for a week, and so the sample size is basically not that large compared to the main panel. Though there are not that, they're still overlapping, but you see there are still differences in terms of duration of sleep, and actually the correlation between self-reported and information from wearables is not that high. I think it's like 0.34, like 3.35. I will come back to it later. What we did, basically, because GWAS on sleep based on UK Biobank, so we couldn't use the work by Sam Jones from Finland. So, but we split the UK Biobank, did our GWAS basically, and we replicated what Tim and Finland found in terms of even genetic correlations, etc. And after we used polygenic score and just feed it into OLS model with lots of interactions and with lots and lots of controls, because you obviously shift work is very endogenous. So, and to control, for collider bias, I've heard of, it's like you either, apart from checking gene-environment correlation, which was not a threat for our case, but you also have to control for a bunch of things, basically to cancel out certain paths. And here it's a bit of SNP heritabilities that I wanted to drag your attention, because I think for me, chronotype and generally sleep is something maybe we don't pay attention that much, at least maybe I just did not. And while even both biologically, it has quite clear, kind of signatures on genome and like certain hormones and stuff. So it's quite interesting polygenic score, and generally quite interesting phenotype and behavior to work with. And I think it's quite informative for social scientists in general. Okay, let's go through the results. So first, here, what I want you to pay attention to, that actually polygenic score of eveningness is not associated with less sleep, which makes sense, because generally being evening person is not good or bad, right? So that was good. You can see on bottom panel, basically interaction terms, that we found consistent, significant interactions for those who are involved in always night shifts, even when you include lots and lots of socioeconomic, like different basically controls, because this is when you also want to torture your model a bit to include lots of controls when it's all endogenous. So that was quite interesting. And further, this is just to exemplify you that it does look like, these inter-individual differences in chronotypes, even what polygenic score is picking up. it does have some, it looks like it does have a bit of protective effect towards these penalties of night shift work. After we looked at the different lengths of work week, and here, interestingly, we found that it's actually, again, those who work always night shifts, and the greatest protective kind of effect observed for among those who work long weeks as well. So basically, obviously, I will be happy to talk because there are certain types of jobs and certain types of contract arrangements, and it's usually some precarious work conditions. So, like there is obviously lots of selection happening, but it does look interesting that in terms of polygenic score and some signal of it is protective power, at least it is more pronounced among those who work long weeks, and this is a visualization of it. And here now we move to to these wearables data, and you see, so obviously sample size is much smaller. What I found interesting that now polygenic score of eveningness actually predictive. So it does look, whatever people are answering in terms of their duration of sleep, there are some, systematic kind of measurement biases happening, or at least measurement technique. We didn't find significant interactions, but in terms of, like sizes and directions, it was kind of in the same vein. So hopefully UKB will be doing more and more wearables data, because it does look, it's important to study at least such outcome as sleep quality and sleep duration. So just to quickly wrap up, yes, we do find that shift workers, they do sleep less, and interestingly, among them, the polygenic score at least has some protective power. And if you translate, minutes of this protective effect towards the percentages of that sleep penalty, it does look, it is one standard deviation basically of polygenic scores has 28% of this protection, which is important. And it points towards that it is, we should look at inter-individual differences, at least when people study shift work, it's there is no such thing as look at what chronotypes are people having and how we different on things like this. So basically, maybe this is a bit of innovation for this session, that it is possible to look with genetic data into something that very hard to observe, and it also very endogenous chronotype, because they are involved in change over time. So you're never sure in terms of direction of associations. At least with polygenic score, you're more or less sure on certain things. And I don't know whether you have the same, but that study was published, and I have a new passion of mine, and I like talking about it. It is about how we measure sleep and what's happening, and why we need polygenic scores, or at least genetic data as well. So I'm entering a bit of assortative mating world, and we were curious whether couples are more similar in chronotypes, because it does, like, there is literature saying that they are. So, and we found, if you look at top panel, so it's phenotypic information, that actually couples are different in terms of their self-reported eveningness. So, and we were like, okay, it's interesting. When we looked at objective data of sleep, we found no, couples are similar in terms of basically wearable-based measure of eveningness. And it does, like, whether you're parents or not doesn't matter. But when we looked at polygenic scores and whether there is any differences, because we're interested, is it initial assortment or maybe it's convergence over time? So we don't see any polygenic scores. So basically, maybe a bit of ad, keep up with our teamwork, because there are some exciting things as well, I think. And I want to I want to thank you for having me and for listening to my bit jet-lagged talk and talking about social jet lags. But I also really, really want to thank these amazing people you see on these slides who are my inspirations and co-authors. Thank you.
Our next speaker is Yeongmi Jeong, who is currently a PhD candidate at University of Georgia, but will be joining Purdue University soon as a postdoc researcher. And she is going to talk about the effect of peers' genetic predisposition to depression on our own mental health.
Okay. Hi, everyone. I'm Yeongmi Jeong. I'm a graduate student in economics department at the University of Georgia. Today, I'm going to introduce my work on the effect of peers' genetic predisposition to depression on own mental health. This is my solo work. Motivation. So depression is a common mental disorder affecting both adolescents and adults. So in the US, in 2020, about 17% of adolescents and about 8% of adults suffered from some type of major depressive episodes. And also the economic burden of it to society is huge. It's about $210 billion each year. And one of the issues with depression in the US is adolescent depression. So adolescent depression is on the rise. So from 2005 to 2017, it's been doubled. And there's broad literature saying that depressed teens are about 3 times more likely to be depressed when they grow up. So understanding the determinant of adolescent depression has an important implication for prevention and treatment. So in this project, I consider peers' mental health as a factor that affects own mental health. The reason is because adolescence is a period in which the influence of peers have more pronounced impact. So I want to focus on peers' mental health. So what I do in this paper mainly is I estimate the short-term and long-term effect of peers' genetic predisposition to depression on mental health by using data from EdHealth. So Kathleen yesterday explained a lot about the data. And in the data, I rely on same gender grade mates average polygenic score for major depressive disorder by exploiting variation within school across grade. And so basically, I'm estimating peer effect. And when we estimate peer effect, there are several econometric challenges need to be addressed. And reflection problem and endogenous group formation are two of those. And reflection problem basically becomes an issue when we estimate own behavior as a function of others' behavior. But in my case, I'm estimating my depression as a function of others' genes for depression. So this is not an issue in my case. And endogenous peer group formation is in general becomes an issue because people people tend to befriend others who have similar traits. So there could be some endogeneity going on. But in my case, I define peers as grade mates. So I try to avoid that issue by defining peers as grade mates. So after doing domain analysis, I was wondering then, what is the mechanisms through which peers' genes operate to have an impact on my own mental health? So I explore friendship, substance use, and socioeconomic status as mechanisms. Yeah, and then literature and contribution. So there are a lot of interesting papers in this literature, but these two strands of literatures are the ones that are closely related to my work. So first, there's a literature of peer effects on mental health, and my work contributes to this literature by using peers' genetic risk for depression instead of observed depression. And also, I focus on the role of adolescent depression in the US other than other countries. And also, in my research, I do analysis for short-term and long-term together, and also I explore a wider range of potential mechanisms than the other papers. Also, the other small but growing literature is documenting the social genetic effects, as Kathleen yesterday briefly explained about this literature. So social genetic effects is basically about whether the genetic makeup of people around me is affecting my outcomes or not. And there are a handful of papers in this literature, and I add to this literature by finding some social genetic effect in the context of mental health. And also my findings suggest a genetic foundation for social contagion of adolescent mental health, which is found in clinical and medical literature. Yeah, so the data that I'm using, as I mentioned, I'm using national longitudinal study of adolescent to adult health. So I'm estimating the results for short-term and long-term. To look at the short-term, I am using wave 1 of ad health, and to look at the long-term effect, I'm looking at wave 4. And then briefly speaking, the genetic data in ad health is collected in wave 4. And the polygenic score that I'm using in this data and project is major depressive disorder polygenic score. And one thing I want to emphasize here is, again, in the last row, as you can see, I used same gender grade mates average MDD score to measure the peers' genetic predisposition to depression. Yeah, here is the summary statistics. So I am presenting the summary statistics by gender. And as you can see here, for most of the demographics and then family and parental controls, the statistics are pretty similar, right? Except for the last 2 rows. So in the last 2 rows, I'm showing depression measures. And as you can see here, for females, depression looks like more prevalent than males. And this is consistent with what the clinical literature is finding. And this is one of the reasons why I'm doing my analysis by gender. And also here is the empirical specification. So I'm running a linear regression here. And Y is the outcome variable, and which is the binary depression variable. And then on the right-hand side, I'm including PGS bar, which is the main variable of interest in this project. So that is the average MDD score of same gender grade mates. And I didn't include myself's PGS score when I calculate that, the PGS bar, the average MDD score, but I include my own score separately. And also, I controlled for individual characteristics and then parental characteristics in the variable X. And also to account for the correlated effect, I controlled for school grade level controls, for example, share of females in the school grade, and then like the share of Black in the school and grade. And also I control for school and grade fixed effects. So that's the empirical specification. And my empirical strategy relies on 2 identifying assumptions. First, there needs to be sufficient variation in the MDD score within school across grades. So what I find out is even after controlling for school fixed effect, grade fixed effect, and school grade level controls, there's a sufficient variation. So the residual standard deviation is about 70% of raw standard deviation, which means that still there is sufficient variation to explore. And then the second identifying assumption to verify is, as I said, there is concern about endogenous peer group formation. So in the US, within a school, assignment into a grade is based on age. So grade mates are assumed to be quasi-random, their assignment. But we want to make sure that that's true in my data. So I try to see whether that is true. So I did several exercise. One of that is balancing test. That is basically running a simple regression of each observed characteristic on the average MDD score, and then to see whether there's a systematic relationship, significant relationship, but I don't find any systematic relationship by running the balancing test. So that I'm saying that this is verified. And also, I did another exercise, which is Monte Carlo simulation by assigning students, randomly assigning students within a school to a different grade, and then see whether the variation in the average MDD score in the actual sample is similar to the one that is calculated from the randomly assigned peer groups. And this one also is satisfied. So I'm saying that two identifying assumptions are satisfied for the analysis. So I'm good to go. So I didn't put the tables here because for the sake of the time. So I tried to just summarize the results that I'm finding. So first, as I said, I'm interested in whether being surrounded by friends who have high genetic risk for depression is affecting my own depression during adolescence. So basically, short-term effect. And what I find in this short-term analysis is that I find increase in one standard deviation, increase in average peers' genetic risk for depression during adolescence increases the depression probability by about 2 percentage points for girls and about 3 percentage points for boys. So basically, there's short-term negative impact, peer effect on depression that I'm finding. And one of the key question of this project is whether that short-term effect persists into adulthood. And I do find, as you can see in the second bullet point, I do find out that, yes, for females, it does persist into adulthood. But it's only for females. But for males, I didn't find much. So what I'm saying is, okay, for short term, yes, there's a peer effect, but in the long term, for females only. So there is some gender difference in the persistence of this effect. That's the main findings of this project. And then I want to suggest some potential explanation for the gender difference in the persistence. So based on some other literatures, so I was thinking, why is that happening? And then I was doing some literature search in the other area. And then there's a theory called the response style theory. So this theory basically says that when males and females face stress or anxiety, they behave differently. So females are more likely internalize by keep on repeatedly thinking about the issue again and again and again without solving out the problem. But males have different tendency. They are more likely to solve out the problem right away, and then forget about it. So that kind of traits can make the difference, gender difference in the persistence of this effect. And also the other reason also could be because male and female, they have different vulnerability, the vulnerability to peer influence is different between male and female. So females are more, there's literature saying that females are more vulnerable to peer influence. So that could be another reason why this is happening. Yeah, so after I find this results, I was wondering then, what is the mechanism? Because the grade mates are the ones are genetically not related to me, but still their genes are affecting my depression, then I might wonder how. So I try to see what could be the mechanisms. So far I explored 3 mechanisms. First, I explored friendship as a mechanism. So the idea here is that if individuals with high genetic risk for depression, if they are somehow more physically or emotionally detach themselves from others, then that might result in worse friendship, which may also deteriorate on mental health. So that was the thought process, and I was trying to see whether that can work as a mechanism. So what I find out is that for females, for all these mechanisms, I didn't find much for males, but I did find some results for females. So what I find out is being exposed to peers with high genetic risk for depression kind of worsens friendship for females. And then for substance use, similar logic is going there. So if there's individuals who have high genetic risk for depression, and then if that person use substance more, then some peer effect going on there as well. And then that might result in some their friends increase in substance use, and substance use is known to be highly correlated with mental health. So that I'm thinking maybe substance use could be another mechanism that the peers genes might operate through. So I checked whether that is also that can also happen. What I find out is for females as well here, being surrounded by peers with high genetic risk for depression increases the likelihood of binge drinking and then smoking. That's the second finding. And then lastly, I was wondering about the persistence of the short-term effect into the long-term effect. And I think socioeconomic status is one of the one of the mechanisms that I can explain this part. So in the short term, I find out that having friends with high genetic risk for depression increases my likelihood of being depressed during adolescence. Then, well, I'm more depressed, and then that might affect my decision to go to college and all these kind of stuffs. And then that kind of important decisions is also can result in having lower SES in adulthood. And then lower SES is known to be highly correlated with mental health. So I'm thinking maybe that's another mechanism worth looking at. So what I find out here is, okay, again, for females, I find much effect than males. So I find some decrease in the likelihood of being employed if I have friends who have high genetic risk for depression. So these three are the mechanisms that I find some significant effects so far. And then, okay, so conclusion. So the main takeaway of this project is that so far, this gives the lesson that genes are an important part of social environment as well. And also in this specific project, I find out that gender difference in the long-term effects on mental health is one of the interesting findings. And there are a lot of things to do in the going forward. So one thing that I can do is exploring more mechanisms. For example, academic performance is one thing that sounds really interesting because the EdHealth has academic performance data that they're providing. And one other thing that I'm thinking of is exploring insulating or protective environments that can mitigate the negative impacts that I'm finding. This could be something like childhood SES or relationship with parents and so on. Yeah, this is all that I prepared for today, and thank you so much.
Our student Jia Miao submitted the following abstract, but he's currently doing an internship at Regeneron, so cannot be here in person. So I'm stepping in and presenting on his behalf. And my name is Qiongshi Lu, and most people call me Q. I'm an associate professor in the Department of Biostats and Medical Informatics here at UW-Madison. And I'm going to talk about gene-environment interaction. My plan is to tell you why I used to think this is a very boring problem and why I changed my mind, why there's a need to reimagine the whole thing and the statistical framework we designed to do exactly that. I have two simple slides to appreciate GWAS. I assume most people here know how GWAS works, but I do have a point to make after that. So the idea of genome-wide association study is incredibly straightforward. You can do GWAS on quantitative trait or binary outcomes, and you analyze the association, quantify the association between each SNP and the outcome of interest, and do it repeatedly for millions of variants in the genome. And in the end, what you get is this type of Manhattan plot, which quantifies the association between each particular SNP and the outcome, and the y-axis shows you how strong the statistical association is. It's a log transform p-value. The higher the value, the more significant the association is. And important note about GWAS is these days, most GWAS is conducted through meta-analysis. So it almost always involves a large number of studies. Each study contributes summary statistics of their GWAS analysis, and someone in the consortium will have to do some kind of average to calculate the average effect size for each particular SNP. And this is what the typical GWAS summary statistics looks like. Each row is a SNP, but you have this type of results for millions of SNPs, and then it tells you the marginal regression coefficient, standard error, confidence interval, test statistic, and the p-value. And then you have this type of results for a large number of variants. The point is, even if you are the first author in the GWAS paper these days, you probably don't have access to all your data. What you have access to is this type of summary statistics. Why do I talk about GWAS? Oh, by the way, after having the summary statistics, you can actually do cool analysis. Yeah? So these days, because of the omnigenicity or high polygenicity of complex trait genetics, you see way too many signals in the Manhattan plot, so people stop bothering labeling them. So, but instead, the focus has generally turned into doing polygenic analysis, right? You can quantify the heritability, you can look at what part of the genome is enriched for the heritability, and you can estimate genetic correlation and see the shared genetics between the outcome of interest with all the published GWAS, and you can calculate polygenic scores, right? Instead of looking at one particular SNP, you aggregate the effect across many SNPs and see whether these scores are predictive. But why do I talk about this? It's over the years, people have developed this playbook for GWAS, and I sort of want to compare G/E analysis that many people are doing to this and to see what's the difference and whether we can learn a thing or two from complex trait genetics or GWAS in general and see how we can use that insight to improve G/E study design. So most people have realized that you really have to do genome-wide analysis instead of candidate gene analysis in human genetics. And big sample size is hugely important. I really have to increase the sample size to be able to say something meaningful, and meta-analysis is a crucial step for that to happen, because no one can have access to individual level data with huge sample size. You have to really, through collaboration, do meta-analysis. And polygenic inference is probably more important than focusing on only top significant findings. I give you a number of examples. And then in this cohort, I think people can appreciate that within family analysis these days is very important because of some complications of confounding and population stratification, sort of mating, etc. So, if you can do within family analysis, that would be good. And then in the general human genetics world, a lot of people care about fine mapping of causal variant and causal gene. But in social genomics observation, I think people generally pay less attention to that. So that's why it's in a different color. Maybe it's less relevant. For social genomics, it's a minor point. So then let's talk about G/E, right? So for a long time, I thought it's a boring problem, because from a statistical standpoint, this is the type of model you have in mind. You have some kind of outcome, you have some kind of G, some kind of E, and the interaction term. You're talking about a linear regression with the interaction coefficient. That is a parameter of interest. Hypothesis testing focuses on that parameter. So, it's not a statistically exciting idea. But Jason, our colleague here, convinced many of us at the UW-Madison that social genomics is cool. So we did some applied project that involve gene-environment interaction. So obviously, we had to look into the actual data. When you look into the data, you realize there's a very interesting asymmetry in this type of analysis, because you're talking about interaction between millions of SNPs and almost always a particular environment. Environment is almost hypothesis-driven, but then you actually have the capability to simultaneously measure a large number of variants. You could argue all the variants in the genome. So it's actually not a symmetric problem. So how do you quantify this type of interaction that involves many, many different genetic variants, where each variant contributes so minimally to the overall interaction? So essentially, you actually need a different type of language to describe, to quantify, to estimate this type of interaction. And people do try. So for complex trait genetics, we now actually speak a somewhat different language, right? I give you a number of examples. People pay less attention to individual SNPs, but people generally talk about heritability, genetic correlation, PGS, which are these polygenic type of measures for how genes impact your phenotype. Can we translate these into G/E world? What does that look like? And people try, some people try to quantify whether the heritability between different environments look the same, right? If the heritability or genetic variance looks different across different environments, that means there is some gene-by-environment interaction. And some people do stratified GWAS in different environments and then estimate the genetic correlation between them, right? If the genetic correlation between different environments for the same trait is below 1, that means they're not identical, right? There's evidence that genetic effect on the same trait is different across environments. This is called differential genetic correlation analysis. In general, this type of analysis obviously leverages the genome-wide data. We're talking about heritability, we're talking about genetic correlation, but it only works for binary environment. You need to be able to do stratified analysis first. And also, in general, this focuses on whether or not there is an interaction or not. And I'll give you examples about additional questions that can be asked. And also, I'm a little bit unclear about what these methods are actually estimating, because the statistical intuition I've always had about gene-environment interaction is something like this. But if you quantify differential heritability or try to compare genetic correlation between environments, it's less clear what parameter you're estimating. It's intuitive, but I struggle to put that type of analysis into this type of linear model framework. And from the previous two talks, you can see that a lot of people try something else, right? It's essentially the polygenic score by E interaction analysis. So instead of looking at one SNP and see how that interacts with the environment, let's construct polygenic score first. Combine evidence across many, many different SNPs, create the score, and see how that modifies the environment effect, right? This is a famous paper in New England Journal of Medicine from actually quite a few years ago. Their story is the environment is the intake of sugar-sweetened beverage, and to see how that contributes to obesity. But then the conclusion is if you stratify people into different susceptibility groups based on their polygenic score for BMI, turns out the environment effect, the intake of sugar-sweetened beverage has the largest effect size in people that are more predisposed for the risk. Right? So you see that the genetic polygenic risk score can modify the effect of the environment and affect, obesity. But inherently, you realize that this is a two-step analysis. To be able to do this, you need a score. Where does the score come from? It has to come from a GWAS. From the first talk, we saw that if your GBI cohort, which is UK Biobank, overlaps with the GWAS cohort, you run into some problems. You need an independent GWAS to be able to create score in your own GBI sample. And also, current polygenic scores are by no means perfect, right? You can demonstrate they are predictive, but there will always be measurement error. So then when you have measurement error, the coefficient will be biased because of that. But I also want to note that just compared to previous examples, this is actually asking a different type of question, right? So the idea is, why do you see gene-environment interaction? It's not just about whether gene-environment interaction exists or not anymore. So this is saying some SNP will modify the environment effect. You actually hypothesize maybe the type of SNP that modify effect are the SNPs that contribute to BMI, or contribute to obesity susceptibility. So people don't highlight that enough in the literature, but essentially this is actually trying to address a different type of question. So the project is motivated by our frustration with how there are multiple types of ways that leverage genome-wide data to talk about polygenic GBI inference in the literature, but it's unclear how to compare them, or whether or not they're estimating the same parameter. And compared to the GWAS playbook, you see that, yes, people are doing genome-wide analysis these days, but if you think about it, actually very few people do genome-wide SNP by E scan, unlike the real GWAS, right? Because a lot of people will say that it's going to be underpowered anyway, but instead people still try to do some kind of polygenic inference by leveraging heritability or genetic correlation of polygenic score, but actually not genome-wide SNP by E scan. Most people still focus on a small data set of GBI inference. No one is doing meta-analysis of GBI across different cohorts. This is a big difference. And of course, a lot of people do focus on within family inference, which is good. So our frustration is that it seems that the GBI world is sort of still in the prehistoric phase of GWAS, and we're trying to translate some of the modern GWAS ideas into how people should do GBI inference, which is why we propose a framework called Pigeon, polygenic gene-environment interaction analysis, in which we propose a very actually straightforward linear model. It's by no means a black box. We're just assuming that the outcome is affected by millions of SNPs through their main effect and one particular environment, and then millions of interaction between each SNP and that environment, right? But then you have way too many parameters to estimate, so we borrow a key technique from heritability estimation literature by assuming these millions of marginal effects and the millions of interaction effects to follow normal distribution. It's a random effect model. So then each SNP follows the same additive effect and interaction effect distribution is ID across different SNPs, but then you have variance component attached to them. So based on this random effect model, you can do two different things. A is to estimate the variance of this guy. This is to say whether or not do I have any evidence there's gene-environment interaction. Based on this framework, to test if there's any evidence for gene-environment interaction, it's about saying whether this term is zero or not, right? If this whole thing is zero, then there's no gene-environment interaction. Based on this model, this is the equivalent to estimating the variance component of this interaction, effect. Okay. So this is the first thing you can do. The second thing, and more interesting thing, is to estimate the covariance between the additive effect and the interaction effect. This is, again, going back to the intuition. If I see a non-zero component here, some SNP give you interaction signals. Why? What are those SNPs? Are those SNPs that increase BMI, right? Do I see the SNPs showing stronger interaction, also giving you larger effect size of BMI or some other traits? This intuition leads to the estimation of the covariance between these additive genetic components and this interaction genetic components. And it doesn't have to be on the same phenotype. You can have the same model for a second trait. You can estimate the genetic correlation between additive effect of the second trait and the interaction effect of the first trait. Okay? So the interesting thing about this is the examples I gave you earlier can be put very nicely into this framework. Essentially, differential heritability in stratified GWAS and also comparing genetic correlation across different environments are trying to estimate the variance component of this interaction term, but neither give you a perfect estimate of that. If you combine these two, you're getting much closer. And also very interestingly, probably more interestingly, the polygenic score Bayes interaction analysis is exactly estimating genetic correlation between main effect and additive effect. In fact, you can mathematically show the equivalence between these two. When you do a PGS Bayes interaction analysis, you're trying to estimate the interaction between the true polygenic score with all the causal effect sizes in your score, and to see how that interact with the environment you hypothesize to be important, right? But empirically, you're actually not doing that. You have to plug in an estimator for that polygenic score. But that interaction between the true PGS and environment, that interaction coefficient, is equal essentially to the covariance between additive effect and interaction effect. So in terms of hypothesis testing, one is these two are essentially the same. You can easily convert one to the other. Okay, so this is essentially a variance component interpretation of the PGS Bayes practice that a lot of people are doing in the literature. But once you realize this is just a variance component estimation problem, estimation becomes a much simpler task. So we actually introduced the LD score regression type of estimator. So essentially, if you have GWAS summary statistics, then you have a G by E summary statistics across many different SNPs. Then you can calculate the product of the SNP level z-scores of these two. You can show that it follows a linear function for with LD score, and the slope actually give you the parameter of interest, which is oracle, the true PGS Bayes regression coefficient. It has a number of advantages because LD score regression type of estimation procedure or genetic correlation analysis is robust to sample overlap, right? It used to be the case I have to rely on independent GWAS, create a score in a second cohort, test the interaction with the environment. Now it doesn't matter. Just have to have one cohort. You can do everything in UK Biobank and perform SNP by E interaction in UK Biobank and also do the GWAS in UK Biobank. You can still estimate the same parameter. And it's unbiased estimation. You don't have to worry about measurement error in the polygenic score. So it's really quite a bit of difference compared to existing study design. This is an illustration of that. It used to be that you need the GWAS to calculate score in a different cohort. Different color means there's no sample overlap. Then test the polygenic score by interaction. Now you don't have to worry about sample overlap. There can be arbitrary sample overlap. You do a GWAS first, calculate the summary statistics, and do a G by E, SNP by E analysis for millions of SNP, get the summary statistics, then test genetic correlation between them to get the same parameter with improved estimation. Simulations always work. You can see that. I'll skip some details because of time, but compared to polygenic score by E analysis, we don't gain a lot of power. It's actually, there's no free lunch. It's similar in terms of statistical power, but you actually get unbiased estimate of the true PGS by E coefficient you worry about. In empirical PGS by E analysis, you get underestimated coefficient because of the measurement error. Right? When there is sample overlap, the PGS by E analysis completely gets ruined. You get hugely inflated type error, but in this pigeon framework, you can still get very good estimates with well-calibrated standard error. We did 3 sets of real data analysis using 3 sets of exogenous environmental exposures. The first analysis is the replication of Patrick's paper. Here it's about how genetics modify the effect of education reform to affect late life health outcome. Long story short, because of time, we basically replicated everything they were able to do, but using a different analysis. They were using PGS by E analysis. Now we're using Pigeon. The things they find to be significant is also significant in our analysis, but then we have improved effect size estimate because we don't have measurement error in the polygenic score anymore. So on average, you see the increase of effect size estimate by maybe 35 to 65%, depending on what you look at. And importantly, we also don't have to worry about sample overlap anymore, right? Because the analysis is done in UK Biobank. If your polygenic score is coming from a GWAS that use sample in UK Biobank, it used to be a problem, but you don't have to worry about that anymore. You get very similar results, even if there's sample overlap between your GWAS sample and the PGS by E analysis. And everything that was null in their analysis remains null in our analysis. So you don't get this weird inflation of type I error. The second analysis is gene by sex interaction on 530 traits in UK Biobank. Here, the quote-unquote environment is biological sex. We want to understand how genetic bases for the same trait differ between different sexes. I will only tell you one story, which is my favorite. The idea is if you look at the GWAS on BMI between males and females, they look different. Genetic correlations below 1, heritability look different. So why? Why do you see this interaction is a key message. What we are able to say is the way it works is to SNP by sex interaction on BMI in UK Biobank. Once you have that summary stats, you test the genetic correlation with everything that has been published in the literature. And the strongest correlation is anorexia. So this is trying to say that anorexia seems to have a much stronger effect on low BMI in women than in men. And anorexia is a genetic trait that partly shows up in the GWAS for BMI between males and females. So this is essentially a point I was trying to make earlier. It's not just about whether or not there is an interaction. It's about, can you say something about interaction? Can you interpret that? And this is a convenient framework that allows you to do something like that in a hypothesis-free manner. And the third example is we also did this in an actual clinical trial. It's a failed trial. I mean, there's different arms of the trial, but a particular aim in that trial was failed. But then we see that the treatment effect is actually modified by polygenic score. In a subset of individuals with higher polygenic score for smoking initiation, this smoking cessation treatment actually do work. You can see significant result. So I'll wrap up. So PGen is a statistical framework to quantify polygenic GBI interaction. It has some superior statistical performance compared to existing approaches, has a number of advantages, and importantly, only needs GWAS and genome-wide interaction study summary statistics as input. We have a bioarchive preprint, and software is available. And this is an important notice. So it's genome-wide analysis. It allows you to do meta-analysis now. This is my favorite part about this because it can potentially change study designs. No one is doing SNP by E interaction meta-analysis because why do you want to do that? When you get the summary stats, what do you do with it, right? You can't just do polygenic score by E interaction, which is what people really want to do. Here is to show that you can do that using the SNP by E interaction summary statistics, which is why probably people should form this consortium to start doing meta-analysis. When you have the interaction statistics, you can actually estimate the same set of parameters that people used to want to estimate. And then a lot of people want to do within family analysis. Just a quick note, you can do that still at the GWAS and GWAS level. If your summary stats come from within family analysis, everything downstream, which I talk about, remains the same. So this is why we think this is a way to reimagine how future GBI analysis should unfold. Current GBI analysis is almost performing a cohort with individual level genetic exposure and outcome data. But this is a demonstration that if you have summary statistics of GBI coming from many, many different cohorts with much larger sample size, you can estimate similar things. And so we make a bold prediction. The future success of complex trait GBI research resides in sharing and meta-analyzing GWAS summary statistics, and method development efforts should focus on techniques that only require summary level data as input. And everything was done by our excellent student, Jiacheng Miao, who's jointly advised by me and Lauren Schmitz, and many colleagues here at UW contribute to the project. I'll stop here. Thank you very much. I think we'll have some time for questions.
Hey, a question for Evelina. You kind of talked about selection into shift work, into night shift work as a sort of a nuisance, and which it can be, right? But it's also interesting. So, do people with the eveningness chronotype, are they more likely to go into occupations that require you to do night shift work?
Thank you. It's a great question. Actually, I'm now thinking maybe I should write a quick paper on that because we didn't find any, kind of correlations. So our gene environment correlation was 0. As I said, having at least polygenic score for eveningness chronotype, not necessarily, good or bad. It looks like everything, the portion of selection goes from social aspect in terms of the conditions you're living, etc. But maybe Thank you for the question. I'm receiving it a couple of times, and now maybe I should kind of write it down. Thank you.
And there was an online question for Pigeon. So the question was, it seems that this type of analysis assumes a similar effect size from environment and genetics on the outcome. And that is not the case. So in typical G/E analysis, obviously you assume there's a genetic effect, environment effect, but I think very few approaches actually assume the effect size to be similar. So just to clarify, that is not an assumption in our framework either.
I have a short question for Yeongmi. So I really liked the paper and both the combination of a research design that might produce causal effects and some of these mechanism questions. So I understood your mechanism through friendship as asking whether people with these type of classmates socialize with their friends more. But I'm wondering if you also asked whether people who have classmates with high PGS for depression don't have friends.
The number of friends.
Yes.
That is actually on my to-do list to look at. So at the beginning, I was just looking at whether, so in the data, there's a questionnaire asking whether you actually spend time with your friend or like visiting your friend's home or this question. So I use those questions to look at the friendship mechanism. And then that's basically the quality part of the friendship. The quantity part will be like the number of friends, right? And in the data, there are in the data, they're asking to nominate your friends. So I think I can count the number of friends and then check the quantity part of the friendship.
Both in degree and out degree. You can ask about sending and receiving friendships.
Yes, like reciprocal.
I have a question for Yeongmi. All the talks were really great, by the way. But a question for you. I know you had a slide about this, but I'd forgotten by the time I was thinking about this. But you're in the regressions that you did, you had controls for school.
Schools,
right?
Yes, like school grade level controls.
Okay, so you were identified off of kind of within school variation, like whether or not you were assigned to a class that had people with higher or lower, relative to the other classes in your school. Is that right?
So in my regression, I'm controlling for school fixed effect, grade fixed effect, and then there's concern about correlated effects. So the effect could be because you are the effects that are specific to that school grade. So to account for that issue, I also control for school grade level controls. So
okay, yeah. And so how many grades? I guess I'm trying to think of what how much. So were there generally multiple? Because you're identified of which classroom you're assigned to of the potential classes, right? How many classes could they have potentially been assigned to in your data? So
in overall, I counted like in my data, it's like genotyped data. So it's not there's adherence data, like in the data there are about 20,000 people, and then again my data set, it's smaller than that because it's genotyped. And on average, there are like 15 or 16 females in each school grade and genotyped grade mates. And then for males, there are about I remember 17 in each school grade. Okay. On average.
And then for the controls, generally when we just have, sorry for a million questions. I'm just trying to understand what you did exactly. And so we generally have PCs, but then did you have some like average classroom PCs as well to control for stratification within a class?
Average classroom. I didn't look at classroom or nominated friend in my project yet because I was trying to figure out the endogeneity in the formation of peer group. So I don't think I checked that. Classroom level average, you mean, right?
Yeah, because I was just thinking that if let's imagine that you just happen to get a draw of people with certain ancestries in your class, and so it could be driven less by having classmates with a genetic predisposition, but more just classmates drawn from a certain population.
Yeah, also I think maybe it could what I can look at is just look at the maximum score and then the minimum score of my class, minimum PGS score, so that whether having the outlier score friends having any impact. I think that's also something that I can do based on your comments. Thank you.
Thank you.
So let's thank everybody